All right, year 12s. We're moving on to the final area of study now for biology. And this is really looking at kind of the use of DNA in scientific applications. So the area of study is kind of called How Do Humans Impact on Biological Processes? And there's a few specific examples that we're going to look at. Now, there's probably some new words in here that you haven't heard before. That's okay. We'll go through them all. What we're going to focus on today is this first point here. So we're going to introduce a few different enzymes that are involved in cutting, pasting, and multiplying DNA. I'll just warn you, I guess, for this topic, the work I'll assign will probably be mostly Ed Rollo questions, just because you'll see as we go through it, it's quite hard to kind of create questions around this, just the way it's structured. And the Ed Rollo questions on this are quite good and very similar to the ones in the exam. So at the end of each lesson, I'll kind of say which questions you need to do. I think it's really important to start with looking at why we do this. So the kind of term we're looking at here is DNA manipulation. And this is basically looking at manipulating or changing the DNA of an organism, and so looking at its genes using bits of kind of technology or biotechnology, which is basically just technology that allows us to work on cells and DNA. Examples of where this is used or why this is used is in pharmaceutical research or creating vaccines, which obviously you've probably heard a lot of this year in the search for creating a coronavirus vaccine. In agriculture, so these techniques are often used to make genetically modified foods, which we'll look at, and also particular animals where we actually not just artificially select, but use DNA manipulation. In medical fields, it's very commonly used for testing for genetic disorders and also treating things. So even things like producing insulin for diabetic people is done using a type of DNA manipulation. And we'll look at that a bit later on in this section. It's also used in forensic fields. So things like testing for evidence. You've probably always, heard about things like getting DNA samples and testing for it, or identifying the remains of a body. Unfortunately, this section has been removed from the study design this year just because of time, but there's some quite interesting stuff in there. All right, I just want to do a quick recap because I think it's really, really, really important that you've got your head around these things that we've looked at the start of this year moving forward. If you don't remember some of this stuff, I really recommend that you go back to the start of your notes and start looking through these things again. All right, so if we look at our DNA structure, we've got a really small section of DNA here. We've got a single nucleotide there. We've got our bases, which are the letters, and our nucleotides. And the bonding of these nucleotides, so kind of between each nucleotide on the side, we have our phosphodiester bonds, which is also sometimes known as the sugar phosphate backbone. So they're found on both sides here. And then in the middle, we have our hydrogen bonds. Sometimes we'll see DNA written really, really simply like this, where we're just looking at the letters. So the bases here, we haven't drawn the whole nucleotide. We're just looking at the bases. And this is kind of what the structure looks like in this topic. Generally in scientific fields, that's how it will be drawn. Just that sequence of letters still represent our hydrogen bonds here. Also remember we've got two ends of our nucleotides and therefore our DNA. We've got the 5' end and the 3' end. And then on the other strand of DNA, it's the opposite. We've got the 3 and the 5 up this way. Remember, a gene is a small section of DNA that codes for a specific protein. So if we have a chromosome that consists of a large amount of DNA, If we unwound that and just look at this one section here, that would be a gene. And let's just use an example. The insulin coding gene makes the insulin protein through the process of transcription and translation. So moving on to transcription, that is the process of a gene being copied into mRNA. Remember, that's really, really important for just making sure we're switching on certain parts of a gene and making specific proteins. I've put this in here just because some of the things in this kind of relate to processes to do with transcription. So it's when you would unwind a double helix and then copy one of them and end up with a new strand. And enzymes, remember, are protein molecules that assist and speed up chemical reactions. The easiest way to recognize enzymes is that their name usually ends in A's. There's not a lot of enzymes that you need to know the name of because there are so many, but there obviously have been some that you would remember. One of them being RNA polymerase, which is the enzyme that helps to make copies of RNA from DNA. Okay, so that's just a recap of some keywords that will start coming up again now. All right, like I said, today we're focusing on enzymes that are used in the process of engineering or manipulating DNA. And there's three main categories that we're going to look at today. One of them are called endonucleases. Remember, it's got A's, it's an enzyme, and these are used to cut DNA. I'm going to look at this in more detail. We've also got ligases. Again, A's for enzyme. These are used to join fragments of DNA or to kind of paste them. So we've got cutting here, we've got joining here, and finally we've got polymerases. So like we looked at before with RNA polymerase, the job of polymerases is to add nucleotides to a strand to create a new strand of genetic material. So that's kind of our copy function. I always think it's a good idea to have a brief summary of what we look at first, and then we'll kind of come back to this. Now I'm going to give you an example again to relate back to as we go through this. So imagine that we want to cut a faulty gene, so a gene that doesn't do its job properly. We want to study that. And to do that, we need to cut it out of the DNA of the person that has this faulty gene. And we're going to, once we've cut that gene out, insert it into a circular bit of DNA called a vector. Now, we'll look at this a lot more later, but just know for now that vectors are bits of DNA that you can insert other DNA into so we can study it. Okay, so this is our human DNA here, and this is the section we want to cut out. That's the gene that we want to look at. We have a vector, which, like I said, is a circular piece of DNA. Now that vector is just going to be in a test tube, in a lab. Okay, it's not inside a living thing. So we've got this vector, and if we want to put this gene inside this vector somewhere, we need to cut open the vector as well. Okay, so we need to cut that open so that we can insert that in here. So you see here, we've cut out a section of DNA from here. We have opened up this vector, and then we can insert a vector, insert the gene into the vector. Okay, hopefully by the end of this lesson, this makes a bit more sense and you can understand the steps of doing that. So we'll start with endonucleases. They're also called restriction enzymes, and the study design has both of these names in it. So you need to know both of these names. Restriction enzymes are a type of endonuclease. These are enzymes that are found naturally in bacteria, but now they're used by scientists, like I said, to cut out fragments of DNA. That's their job. That's just a picture of what an endonuclease might look like. This is kind of what we mentioned before, but if we want to study a certain gene, we need to cut that out of its original big strand of DNA. And that's where endonucleases or restriction enzymes come in. I've just included this because you will see examples of names of restriction enzymes. You don't need to memorize any of them, but they will come up, and it's important that you kind of can recognize their names. So they're named after the particular bacteria that they're found from. Okay, so like I said, you don't need to memorize these, but it will help. These are the names of a few restriction enzymes. There are lots and lots and lots of them, but the name derives from the type of bacteria it comes from. So BAM comes from this Bacillus amyloid. I'm not even going to continue trying to say that. This is a really common restriction enzyme here, ECORI, which comes from the E. coli bacteria. Don't worry about this just yet. We'll come back to that in a minute. Okay, but that's what the names of them look like. Now, specific restriction enzymes are used to cut at specific sections of DNA. And we call these sections of DNA recognition or restriction sites. So what we mean here is if we want to cut this gene out here, if we want to look at this gene, we need to be able to cut on either side of that gene to remove it from that whole piece of DNA. It's really important that we don't cut halfway through the gene because then we're going to miss some of that gene. So a particular spot in the DNA that a restriction enzyme can cut is called a restriction site. A restriction site is a very small sequence of DNA. It's usually 4 to 6 nucleotide bases long. And they're often, or usually, they're in a palindromic pattern, which means that they are the same forwards as they are backwards. I'm going to show you a couple of examples of that here. Remember, DNA is double-stranded. So I've got one strand here. We'll look at what the other strand would be. So ECORI is a restriction enzyme. And this here, these bases are the restriction or the recognition site that this restriction enzyme will cut. If it encounters a DNA sequence like this, it will cut it there. Where the palindromic thing comes in is, if we look at these patterns, we've got G, A, T, T, C. The complement of that is C, T, T, A, G. So it's the same this way as it was that way. GAATTC, GAATTC. Okay, we find that the same with this one. If you turn it upside down, it will look the same. This last one is a small restriction site for a particular restriction enzyme, but again, it's still a palindrome. We end up with T, C, G, A, T, C, G, A. Okay? Now, when a specific restriction enzyme binds to its specific recognition site, it will cut at that site. The way it does this is by cutting or cleaving the phosphodiester bond on both of those strands. I'll come back to that in a second. So remember, we've got our hydrogen bonds in the middle. We've got our phosphodiester bonds between each nucleotide here. These are the bits that get cut, okay? Not the hydrogen bonds, but it's important to know that hydrogen bonds are unstable. And if we cut here and here, for example, this bond is weak and they will separate, okay? Restriction enzymes cut at a certain point on that recognition site. So this is what I mean by how DNA will be kind of shown to you in these types of questions. It's just the letters. So first we've got this restriction enzyme, specific restriction enzyme. If that restriction enzyme is put into a test tube with DNA and it comes along this particular sequence of DNA, this recognition site, it will make a cut here on the top strand and here on that bottom strand. So it'll cut right through the middle. Okay, you see that again here with this other restriction enzyme. It'll find a different restriction site and cut right through the middle. Then there's a couple of funny looking ones where it will cut on one side here and cut down here on the other side. So it's cutting between the two G's on both sides. We'll come back to that in a second, but just take note of those two different shapes. Because of the way it cuts, there's two different kind of shapes of the strands that we will end up with. One is called blunt ends. So like in this top one, if we cut right through the middle here, we will end up with the DNA in two strands, strand 1, strand 2, with a really blunt ending. Okay, it's a straight cut. The other one, the other shape, is called sticky ends. So like this one here, if we cut here and cut here, and then those hydrogen bonds in the middle just break, it ends up getting separated like this. So we still end up with two strands, one strand here, one strand here, but we've got this overhang, and that is called sticky ends. It is really important. It comes up all the time in exams, and we'll come back to that a little bit later. All right, this is another important thing to note. In a section of DNA, the same recognition site might show up multiple times, because remember, it's just a sequence of between 4 and 6 bases. So there's a good chance that that will show up multiple times. If we have a bit of DNA and it's got that recognition site multiple times and we stick some restriction enzymes in there, they will just cut anytime they see that site. So the first example here, if we have got a straight bit of DNA, a linear piece of DNA, and we have a restriction site here for the EcoRI restriction enzyme, it'll cut there and we'll end up with 2 strands of DNA. Okay, so it cut in the middle, we've got 1, we've got 2. Okay, that's pretty straightforward. That makes sense. If you cut in one spot, you'll end up with 2 pieces. Now let's say there's 2 restriction sites for the same restriction enzyme. So we put the restriction enzyme in, comes in, it cuts here, then it keeps going, it finds this one, it cuts here as well. Now we've got 3 pieces. We've got that piece, this middle piece, and we've got a 3rd piece. It's a little bit different if our DNA that we're cutting is circular. And this is, if you remember back to the example we looked at, if we are looking at cutting a vector, which is a circular piece of DNA, if we have one restriction site here and we cut here, we're not technically going to end up with 2 pieces because it was a circle. If you had an elastic band and you cut it, it's just going to open up the band, which is what would happen here. We have cut this spot, but we haven't cut it into 2 pieces. If we had 2 restriction sites and it went through and cut both of them, we would end up with 2 different pieces. So this long piece here, and then that piece here would cut it into 2 different sections. This is an example of a question from a past exam that kind of looks at this. So this shows us for a particular plasmid, which is just a circular DNA. This is like a map, really. It's a map of all the restriction sites for specific restriction enzymes in a bit of DNA. Okay, so these are the restriction enzymes and these are their sites. Now it says we've got 3 different restriction enzymes in this map. And it says if the plasmid was incubated, which basically just means mixed, if we mixed this bit of DNA with this restriction enzyme, how many pieces of DNA will we end up with? Okay, so what you could do is cross out these restriction enzymes because the question's not asking about them. We don't care about these restriction enzymes. We're only putting this one in there. So it'll go through and it will cut here. It will keep going. It will cut here. It'll keep going. It'll cut here. And it will keep going and it will find this one. It will cut here. So we will end up with 1, 2, 3, 4 pieces of DNA. Right, so we've looked at endonucleases, or as we now know, are called restriction enzymes, and their job is to cut bits of DNA. Now we're going to have a look at ligases, so the job of pasting bits of DNA. So these are enzymes that join two fragments of either DNA or RNA together, and based on that, they're either called DNA ligase, which joins two pieces of DNA together, or RNA ligase, which joins two pieces of RNA together. They do this by reforming those phosphodiester bonds that were broken or that were cut by the restriction enzymes. So we've got our 2 strands here, one here, one here. DNA ligase will rejoin these bits here, those phosphodiester bonds, and we will end up with now just 1 strand of DNA. We call this recombinant DNA because we have basically recombined 2 different strands of DNA together. So this here is a recombinant DNA. And this is kind of like that example we looked at the very beginning. So we've cut open our plasmid, we've cut this gene out that we want to look at, and DNA ligase will now join the phosphodiester bonds of the plasmid and the gene together until we now have 1 recombinant DNA that's a mix of those 2 original pieces of DNA. A couple of important things about ligases. So firstly, they don't have specific recognition sites. So we don't need a different type of ligase for every different sequence of DNA that we have. The important thing is that if we have DNA that was cut with blunt ends, remember that's that straight cut, it can only be joined with another piece of DNA that has blunt ends. It's kind of like a puzzle. If this was cut here, it's not going to fit up neatly with this side here. Okay, so blunt ends can only join with blunt ends. The problem with blunt end restriction enzymes and then trying to join them together is that because there's no overhang, there's nothing to kind of hold these together while DNA ligase joins here and here. So blunt end restriction enzymes are not actually used that often in science because they don't work very well, because they might meet, but while DNA ligase is trying to join them together, they might just separate again. On the other hand, sticky ends can only join with DNA that has been cut with the same restriction enzyme. So if we look at our first strand here, it was cut here, we've got sticky ends. The exposed bases we have here are A, T, and T. And you know with DNA that they will only join with their complement. So T will only bind with A, and A will only bind with T. So because this bit of DNA was cut with the same restriction enzyme and has the same nucleotides free, they will bind together. If this other piece of DNA was GCCT. It wouldn't bind because this G is not going to like this T. They will be repelled by each other. Okay, so sticky ends can only join with DNA that has the same sticky ends. Again, it's like a puzzle. This is almost like a puzzle and a magnet together. The bases have to be complementary. And the other advantage of this is that while, I'm just going to draw this here, while DNA ligase is doing its job, these bits of DNA will hover, and the hydrogen bonds, even though they're weak, those hydrogen bonds will be enough to kind of hold it together briefly while DNA ligase joins here and here, and we end up with our nice recombinant DNA. So back to our original example. We've got our DNA that we want to cut out of here, and we want to open up this vector. We want to put this bit of DNA in here and end up with our recombinant vector. So we cut the gene out with a restriction enzyme. Restriction enzymes cut DNA, and we make sure that those restriction enzymes are on either side of the gene. We cut open our vector with the same restriction enzyme. Okay, so remember what we just said about them having to be complementary. So we've got these two ends here that because they were cut with the same enzyme, they're going to meet really nicely. And when they hover in there, DNA ligase joins those complementary strands together, and we've ended up now with a recombinant DNA. I want you to pause this here and jump in. I posted a link to a YouTube video. It only goes for about 2 minutes, but it's just a good narration of these enzymes and how they work. Okay, we're almost done, I promise. The last bit we're going to look at really briefly, it sets us up for the next lesson, are polymerases. And these are enzymes that copy or build DNA or RNA. And you've seen these before with transcription. They are enzymes that add nucleotides to DNA or RNA. We've looked at RNA polymerase quite a lot, and we'll look at DNA polymerase in the next lesson. The importance of this in terms of science is that it allows us to copy sections of DNA. If we have one single template strand, so let's say this is a template, we've removed a gene that we want to study, we can separate those strands, and then we can use this strand to build more DNA. Okay, so this is DNA polymerase. It's moving along this way, and it is getting complementary nucleotides, adding them to this template strand until we end up with an exact copy. The other important thing, again, we'll look at this in the next lesson, but for polymerases to work, they need things called primers, which is basically just a starting point. They need to know where they need to start copying from, but again, we'll look at that more in the next lesson. Okay, so our enzymes used in DNA manipulation. We have endonucleases. They are our molecular scissors. They cut DNA. We have our ligases that rejoin bits of DNA. And we have polymerases that build new bits of DNA or RNA. I know that's a lot of content, and it's tricky stuff to learn online. So what I want you to do is to go into Edrolo. I want you to have a go at the first 9 questions in 15A. Okay. If you are using your textbook, I highly recommend just having a quick read through the 2 pages before that and answer those questions. After you've done those questions, I want you to complete my quiz, which is purely just for feedback, so I can see what you understood, what you didn't understand. The reason I want you to do that after you've done the questions is you can see, did you get those questions right? Did you think you knew what they were, but then you didn't know what they were? so I can look at going over some topics again. All right, I will speak to you all again later.